Hi guys, welcome to another lecture in the series Medicinal Chemistry 2, and today we're going to be talking about antineoplastics. So antineoplastic agents describe the chemistry, the use, metabolism, and adverse effect profiles for the alkylating agents, the antibiotics, the natural products, the anti-metabolites, and tyrosine kinase inhibitors used in the treatment of cancer. Now, this is very much going to be a very short overview of anti-neoplastic agents because we are not able to go into every single category of agents and speak at length about them. So this is basically going to be kind of an overview. So cancer, according to the American Cancer Society, is defined as a group of diseases characterized by uncontrolled growth and the spread of abnormal cells that, left untreated, may lead to death. Um, Cancers most commonly occur in the breast, the prostate, the lung, the colon, and the rectum. So I'm pretty sure that we're familiar with cancer. And we may or may not know somebody related who has it. Just to define another term, neoplasia. Related to this definition is the term neoplasia, which is the uncontrolled growth of new tissue, the product of which is known as a tumor, and these tumors may either be malignant or benign. So malignant tumors have the capability of invading surrounding tissues and moving to distant locations in the body in a process known as metastasis. So you might have heard about that before. Characteristics that benign tumors do not possess. Right, so here we're making a distinction between a malignant and a benign tumor. So in terms of treatments, which we call antineoplastics, treatment of malignant tumors or cancer has generally involved initially surgical removal followed by radiation and or chemotherapy if necessary. So in those cases where complete surgical removal is not feasible, radiation and chemotherapy become the only available options. The term chemotherapy refers to drugs that are used to kill cells and includes both antibiotics and agents used in the treatment of cancer, but is often used to refer exclusively to anti-cancer agents known as antineoplastics. All right, so you may or may not be aware with the challenges associated with antineoplastics. Well, traditional chemotherapy has been based on the principle of selective toxicity. So, in other words, you have some cells and you want the therapy to selectively to choose the cancer cells over the normal cells. However, this has been difficult to achieve in the case of cancer cells because these cells utilize the biochemical pathways used by normal cells. So, in many cases, the agents have attempted to exploit the increased proliferative rates of cancer cells compared with normal cells. So this has been difficult to achieve even in a relative sense because of the fact that not all normal tissue is slowly proliferative, and conversely, not all cancer cells are highly proliferative, right? So it's hard sometimes to distinguish between normal cells and cancer cells, so this is the challenge. Um, some common characteristics of neoplastic cells, well, they are persistent proliferation and immortality, differentiation, as well as invasive growth, formation of metastasis, and sustained angiogenesis. All right, so speaking about cancer etiology, the abnormal behavior of cancer cells results from DNA alterations. So, malignant transformation results from a combination of activation of uncut genes and inactivation of tumor suppressor genes, right? So, we do have these tumor suppressor genes. So, these genetic alterations are caused by one, chemical carcinogens, so such as cigarette smoking. Two, pathogens, so or H. pylori or hepatitis C virus. Radiation in the form of X-rays, hereditary factors, and drugs such as estrogen. So these can cause genetic alterations which may or may not lead to cancer. So here we have hereditary chemicals, viruses, radiation, and basically it's possible to cause DNA alteration, which would therefore lead to cancer. All right, so in terms of treatment modalities, surgery, so removal of those solid tumors, radiation, elimination of solid tumors, drug therapy, so disseminated cancers, some localized tumors, adjuvant to surgery, and radiation. So you might take the drug in addition to surgery and radiation. So, goals of therapy. Right, so really for our antineoplastic, we want a cure, right? So, for example, in Hodgkin's disease, we want to prevent the reoccurrence of the cancer, or we want prolonged survival. And in case we're not able to cure it, we want to be able to be relieved of some of the symptoms. So the outcome of therapy depends on the general health of the patient, responsiveness of cancer type in terms of the size, location, grade, etc., and of course, drug resistance. All right, so in terms of drug selection, combination therapy is more effective than single drug therapy. That causes maximum cell kill. It reduces injury to normal cells. It can also slow or prevent development of drug resistance. Right? So, criteria of drug selection for combination therapy. Well, you want drugs that have different mechanisms of actions. That is, they act at different cell cycle stages. You want different toxicities and a decrease in overlapping toxicities, right? So at the end, I think is the last slide, we're going to talk about the toxicities. You want different toxicities. And each drug is effective by itself, right? So these are the criteria of drug selection for combination therapy. In terms of tumor staging, most commonly applied staging system for solid tumors is TNM, which is tumor nodes metastasis. classification. This is the most, TNM is the most commonly used staging system for solid tumors, or classifying them, where a numerical value is assigned to each letter to indicate size or disease extent. So this is a TNM system. So for T, that refers to the size of the primary tumor. So it can be 0 to 4. So if you have a number 4, if it's a T4, then it's a large size tumor. In terms of N, N refers to the extent of lymph nodes involvement, and that goes from 0 to 4 as well. So if N is 0 or the subscript is 0, then no lymph node disease. For NX, lymph nodes not assessed, right? So it doesn't fall in 0 to 4, so it's not assessed. For M, it refers to the presence or absence of distant metastasis, and that's 0 to 1. So M1 means distant metastasis, M0 means no distant metastasis, or MX means distant metastasis not assessed. So if this is your staging T2 N1 M0, then this means that you have a moderate size tumor with limited nodal disease. So that's 1, and no distant metastasis. All right, so that is basically the TNM system. All right, so let's talk a little bit about the cell cycle. So we have our G0, which is our resting phase, and then that goes to G1, which is the synthesis of components needed for DNA synthesis. And then the S phase is synthesis of DNA, and then the G2 phase is synthesis of components needed for mitosis. And then you have M phase, which is mitosis, and then the cycle continues, resting, and all that, right? So you have cell cycle phase specific or phase specific drugs, and you have cell cycle phase non-specific or cycle specific drugs. So we're gonna be talking about both. The cell cycle phase specific drugs must be present for an extended time. They're given by infusions or infrequent small doses. So the G1, based on our cell cycle here, so we're looking at the different cycles, and for G1 phase specific drugs, we have an enzyme, asparaginase. For S phase specific, S phase, a G1 is our synthesis of components needed for DNA. S is a synthesis of DNA. So S phase specific drugs, we have a number of them. Fluorouracil might jump out at you, or any of these. These are our anti-metabolites. And then we have our G2 phase specific. So G2 would be synthesis of components needed for mitosis. So our bleomycin, which is our anti-tumor antibiotic, would be effective there. And we're going to look at our Vinca analoids, which is vincristine, and vinblastine, which is mitotic inhibitors, right? So these are cell cycle specific drugs. So we'll be looking at vincristine and vinblastine here. All right, so we have a question. If a tumor is categorized as T4, N2, MX, explain what this means to a patient. Explain the diagnosis. All right, so if you had T4, means size tumor, a large size tumor, and N2, that the lymph node contains cancer or limited nodal disease, that would have been correct. And MX, of course, would mean distant metastasis not assessed. All right, let's talk a little bit about Periwinkle, right? And you might be wondering where this comes from. This is where, we get our Vinca alkaloids. So, periwinkle, the best known medical agents from rosy periwinkle, arose from investigation into the plant's use in Jamaican folk medicine. Known as periwinkle tea in Jamaica, the well known folk use of rosy periwinkle led researchers to delve into its natural chemical properties in the 1950s. Scientific analysis of rosy periwinkle led to the discovery of two previously unknown compounds, that is, vincristine and vinblastin, which have been subsequently developed into potent medicines to save lives from leukemia and Hodgkin's lymphoma, respectively. So here we see that natural products, persons used to use it in folk medicine here in Jamaica. And, researchers came, isolated these two compounds, and we have our Vinca alkaloids. The Vinca alkaloids are extracted from the leaves of the periwinkle, and the scientific name is Catharanthus roseus, which is usually italicized, and were originally investigated for their hypoglycemic properties, but later found to possess antineoplastic actions. So the Vinca alkaloids. The alkaloids are composed of a catharanthine moiety containing the indole subunit. So we have the indole subunit circled here, and the vindoline moiety. So we have the vindoline. Well, first we have the catharine subunit, and we have an indole as being a part of that. And then you have your vindoline subunit seen here. So again, it's composed of a catharanthine moiety containing an indole subunit and a vindoline moiety containing a dihydroindole here subunit joined by a carbon-carbon bond. So this bond here, carbon-carbon bond, is what joins the catharanthine subunit to the vindoline subunit. All right, so another type of drug we mentioned previously, let me go back to it, cell cycle phase specific drugs. Now we're going to look at cell cycle phase non-specific drugs, or cycle specific, and they act during any phase, including the resting phase. So more toxic to proliferating cells than resting cells. All right, so these are some examples of these types of drugs, and there are alkylating agents. So we'll be talking about cisplating, and you have your anti-tumor antibiotics in doxorubicin. All right, so we have another question. Draw the cell cycle phases. So I hope you can remember it. I've looked at it a number of times, so just see if you can remember the phases. All right, so if you had your resting phase followed by your G1, which is your synthesis of the components in the DNA, G1 phase, and your S phase, the synthesis of DNA, then your synthesis of components needed for mitosis, and then mitosis, then you are indeed correct. And we need to know that so that we can remember, for example, the phase specific drugs, where they act. So I did mention that we're going to talk about cisplating, which is an alkylating agent, and they're a class of drugs that are capable of forming covalent bond bonds with important biomolecules. So, the major targets of drug action are nucleophilic groups present on DNA, especially the 7th position guanine, seen here. However, proteins and RNA, among others, may also be alkylated. So, alkylation of DNA is thought to lead to cell death, although the exact mechanism is uncertain. So, we have new DNA here, we have the nucleophile and the hydrogen, and we have Rx, right? So, normally when we see Rx, we know that R is possibly some type of alkyl or something, and or X is usually some leaving group, right? So it might be a halogen. Then you have alkylation, so essentially replacing that hydrogen with R, whatever R is, and loss of this leaving group, which is X here. So this is literally how these alkylating agents work. So for cisplating in particular, this is the structure of cisplating. So we have platinum in the middle, we have ammonia, and then we have two halogens in the form of chlorine. So essentially what happens in terms of the mechanism of cisplating activation and formation of DNA adducts, we have hydration occurring, so water molecule replacing one of the chlorides, and then after that the nucleophile of the DNA now replacing the water molecule, and that literally happens a second time, right? So replacing the other halogen seen here. Right, so that's basically how it works. And as we said, the alkylation of DNA essentially leads to cell death, and even though that mechanism is unknown. So I did mention that a lot of these antineoplastic have associated toxicities, and just to mention the unique toxicities of some of these agents. So we did talk about vincristine and vinblastine, so it has neurotoxicity, and cisplatin, ironically. And cisplating as well has nephrotoxicity, and we can see a variety of toxicities there. So remember what we said earlier in terms of drug combinations. You want to choose a combination that does not have similar toxicities, right? So it's important. That's why I mentioned these unique toxicities. All right, so finally, I want for you to answer for class tomorrow. Describe the mechanism of action of cisplating, right? So you have reached the end of this video. Thank you so much for watching, and see you next time.